Hello, everybody, and welcome to the last lecture on plants. For those of you guys who indicated in the survey that you were interested in plants and you found them interesting, you were interested in plant biology, sorry, we're reaching the end of this section of the class. And for those of you guys who put you really don't care that much about plants or you don't like plants in the survey, congratulations, you've made it to the end. So in this lecture, we're going to cover plant growth, reproduction, and environmental responses in far more detail than we've covered in the previous lectures. And I would also like to make some clarifications and some changes to some things I said in previous lectures regarding double fertilization. So in this presentation, we'll talk a lot more in detail on double fertilization, and I want to make clear some things that I misspoke about in the first lecture to make sure that they're being correct and being conveyed properly in this lecture. So this is chapter 20 of your textbook. Let's get started. So let's begin with reproduction. Plants, for the most part, undergo sexual reproduction, and for the next couple slides, we're going to talk mostly about that. However, plants can also undergo asexual reproduction, and this is also called vegetative reproduction, and it generally occurs, it's more prevalent, I'll say, in regions where it's difficult for the individuals of a species to reproduce sexually. So, for example, if you have a particular species of tree and the individuals within that species are far separated, so far that it's less likely that the gametes from one plant will be able to successfully be moved to another plant via pollinators or wind or something like that. In those cases, in order for the plant to reproduce effectively, they may opt for asexual reproduction rather than sexual reproduction, so that they're able to reproduce without the gametes or the assistance of another plant. And the daughters that result, or this offspring that result from asexual reproduction, are clones. So they're genetically the same as the parent plant, and they are a result of mitosis, not meiosis. So in meiosis, to give rise to a offspring, you usually have to have two haploid gametes will meet to form a diploid zygote. In the case of clones or in asexual reproduction, it undergoes mitosis. So a somatic cell is basically differentiating into the offspring. And you can get clones from clippings. You can get clones from essentially small pieces of the plant. They can be from the roots, the stems, or the leaves, and that can give rise to a new plant. So using this image as an example, this plant has all these little kind of like buds on the ends of here on their leaves. Each of these buds is actually a clone of the parent plant. And once they fall off, or they are blown off or knocked off, and they get to a substrate in another location, they will form their own roots and leaves and stems and become their own plant. But they'll be genetically the same, they're genetically identical to the parent plant they derive from. You can also do this as an experiment at home if you ever want to. You can take certain plants and cut them in half, and then put the cut side in some water. So I think sometimes you do this in school with like maybe onions or potatoes or carrots and things like that. You can dip the cut end of the carrot in a glass of water or in some soil, and you'll start to see roots will form. And so this is one way, another way of cloning, and it's actually one of the most common ways in agriculture that we get the food that we like. So commonly we like foods that have no seeds, at least in America we do. And in order to get those, a lot of times what farmers will do, they'll take clippings and cuttings from the parent plant and then plant them somewhere else, so you can get a offspring that is genetically the same as the parent plant, so they'll also be seedless. And you basically just keep doing that over and over and over again, and so we end up with a lot of clones of the same seedless plant that will make fruits and veggies that consumers generally like to buy. And this clones, as I mentioned, is a result of undifferentiated cells. So these cells in the parent plant are undergoing mitosis, but they're undifferentiated cells. So they're not already determined to be xylem or tissue or phloem or anything like that, or a root cell. They are totipotent, which means they can become any type of cell the plant needs. And in this case, the plant wants to undergo asexual reproduction, so that those cells will start to make a new offspring. They'll differentiate to make offspring. So there are some advantages of undergoing asexual reproduction. It's energetically efficient because you don't need to make reproductive structures. It is energetically expensive for plants to make flowers and fruits. It requires a lot of energy on the plant's part. But if you don't have to make those structures, if you can reproduce asexually, then that's a huge benefit. Also, it allows for them to reproduce more fast than if you undergo sexual reproduction, which in turn increases their fitness, right? So remember, we talked about fitness is all based on your reproductive success compared to other individuals within the same species. And so if you're able to reproduce faster, that makes you more fit. And in this case, in asexual reproduction, it's faster because you don't need to wait for the gametes from another individual to come and fertilize your gametes. You can just reproduce. So it can be very quick in comparison to sexual reproduction. Also, it's able to preserve advantageous alleles. So when you have genetic recombination that occurs after the haploid gametes meet, sometimes you end up losing certain alleles in the gene pool because that particular sperm didn't have those alleles, and the sperm that did went and died off. So that particular advantageous allele may or may not be lost depending on if those particular sperm having that allele are able to make it to an egg and produce an embryo and a zygote. In asexual reproduction, you don't have to worry about that. All of the genetic material from the parent plant is given to the offspring, including all of the advantageous genes and alleles that allow for that parent plant to survive and thrive and make it to maturity and reproduce. So those are some advantages to asexual reproduction. There's a huge disadvantage to it. There's little to no genetic variation when you undergo asexual reproduction. That's because everything is a clone. There is no change in the gene except for when you get mutations and things like that. Other than the mutations, they're all basically the same. And this leads to plants that are more susceptible to various calamities like disease and natural phenomena and things like that. Disease is a huge problem in agriculture because what happens is, like I mentioned, in agriculture we have a lot of clones. Well, they're all genetically similar. That means they're all represent similar susceptibility to like fungi and bacteria. So in places like Florida and California, they've been experiencing issues with a fungus that attacks citrus trees. If you have a grove of citrus trees that are all genetically slightly different, some of those trees will be more resistant to the fungus infection than others. But if they're all the same and they're all susceptible evenly, then those fungi kind of run rampant through those orchards and decimate the trees. And that's essentially what has been happening over the last few years, is this fungus has been just wiping out whole groves of trees for apples and plants and limes and all types of different type of fungi attack different trees, but particularly citrus trees have been hit really hard by this fungus. So this is kind of a big disadvantage because you could end up being completely wiped out, your entire species wiped out, if you only rely on asexual reproduction. So sexual reproduction is favored because it gives you genetic diversity, which allows the organisms to be able to adapt better to change and be more evolutionarily successful in the long term. But if you have to, a plant will use asexual reproduction if that is the best method for them to at least reproduce to some extent. So now we're going to take a little minute to review the anatomy of a flower. I know we talked about this, but a review can't hurt. So we have our sepals, which are our modified leaf-like structures at the base of the flower, and they're primarily responsible for protecting the flower before it has matured and bloomed. So when the flower is in a bud, the sepals are the leaves that cover the bud and protect it from any environmental damage or damage by other organisms or predators and things like that. And they later, when it opens up, they become the sepal at the base of the flower. And then we also have our petals. I think everybody knows what a petal is. They're the beautiful part of the flower that we're usually attracted to. They come in a variety of colors, shapes, and sizes, and they're all specified to attract specific pollinators. So the beauty of a flower is not for us, it's not for anything that's not their pollinators. We benefit from that, how beautiful they are, but really every single flower has specific petals to attract whatever pollinator is, or pollinators, they can have more than one, that will help to transfer their gametes from one individual to another of the same species. Then we have our stamen, which are our male reproductive structures, and they include the filament, which is this stalk-like structure that holds the anther in the air, and then the anther. And the anther is the site of pollen grain production. And pollen grains, we're going to talk about everything that happens in the anther and pollen grains and all the stuff in a second, but the pollen grain are the structures that contain the sperm. Pollen grains are not the sperm, they just contain cells that give rise to sperm later on. And then we have our carpel, which is our female reproductive structures, and they include the stigma, which is this sticky padding at the very top of the reproductive structure, a style, which is this neck-like structure that leads to the ovary, and the ovary is this entire large rounded structure at the base of the flower. And then the ovary contains the ovule, which is this structure here in this image, and the ovule is the site where the egg is produced. The ovule also later becomes the seed, and the ovary later becomes the fruit in fruiting plants. So keep those things in mind, and we'll talk a lot more detail on those in a second. So plants that contain both the male and female organs within the same plant are called hermaphroditic. Another word you might hear is monoecious, and that just basically means both the male and female gametes and reproductive structures are located within the same organism. So in this case, it's very easy to see. In a single flower, we have both our male and female reproductive organs, and our male and female gametes are located within the same flower. But a hermaphroditic plant can also be a plant that has its male and female structures on different parts of the plant. So if you have, and we'll talk about this in a minute, like corn. Corn has its male and female structures on the same corn stalk, but just in different sites within that stalk. And those are still considered hermaphroditic. So now we're going to get into the nuts and bolts of how pollen grains are formed, how the egg is formed, and how double fertilization occurs. And during this kind of next few slides, take note of all the key details, but also there are some things that I mentioned in the first presentation that are clarified here or presented more accurately in this presentation. So keep note of what occurs, the steps that I mentioned, and all the things that occur during double fertilization in this presentation rather than the first one. So let's get into it. In pollen grain formation, pollen grains are the male gametophyte. So remember we talked about sporophyte is the diploid structures of the plant, the diploid parts of the plant. And then the gametophyte is the haploid part of the plant. And part of the life cycle. And so the male gametophyte is the pollen grain. The pollen grain is watertight and it's sticky, so it's very good at being protected from a lot of different elements, and that sticky nature helps it to stick to the stigma. So the stigma is sticky, but the pollen grain is also sticky, and part of the stickiness involves surface proteins that will bind to elements on the stigma and help it to stick. So what are the steps of making pollen grains? So we start with a diploid microspore mother cell. And that would, in this image, that would be this large cell here. This cell is diploid and it is derived from the anther. And so the anther has a couple of chambers called pollen sacs, and specifically they have 4. So if you were to actually cut an anther this way, down this plane, and you kind of looked at it head on, you would see kind of 1, 2, 3, 4 type of like holes or chambers, and then it will kind of look bumpy like this. Each of those chambers is a pollen sac, and it contains a ton of these microspore mother cells. And then the spores are not gametes. So I'm just going to reiterate that, especially because they're diploid. So we haven't got to gametes yet. We're just in the process of making those gametes. So this microspore mother cell then undergoes meiosis to form microspores. So each microspore mother cell will divide via meiosis to give you 4 microspores. And these are all haploid. So you can see here's 1, 2, 3, 4. Okay. And each of those microspores will then undergo mitosis to form 2 haploid cells within a pollen grain. So each of these pollen grains here, which they're showing the pollen because they have that outer coating that would be their sticky surface, contains 1 cell that will become the sperm later on after pollen tube formation, and another cell that will be responsible for forming the pollen tube. So once the pollen grain sticks, one of those cells will start to make the tube. The other one of those cells will travel down the tube, later divide to become the sperm, 2 sperm. And they divide via mitosis. On the female reproductive structure side of things, we first have to have our ovule. So the ovule is formed by cells within the ovary that undergo differentiation to kind of form these chambers that will be the site of where the egg is produced and the site of fertilization. So the ovary, just as a reminder, is this large kind of bulbous region there. That whole thing is the ovary, but the ovule is just this chamber within the ovary. And a single ovary can have 1 or more than 1 ovule, depending on the species that of plant it is. And the ovule has a couple of different cell types. We have our outer coat, our protective outer coat, which is this here, this kind of light green. And then this cell in the middle is our megaspore mother cell. And that megaspore mother cell is what will eventually give rise to the egg. So looking at how the egg is produced, we start with our megaspore mother cell, which is diploid, and it undergoes meiosis. And when it undergoes meiosis, it will result in 4 daughter cells. And 3 of these daughter cells, so these smaller ones in this image, 1, 2, and 3, will then go on to differentiate and do other things. However, one of those cells, our star, will then go on to give rise to the embryo sac. So this 1 megaspore cell, so this one that goes on to give rise to the embryo sac, will then undergo mitosis, and it will result in several rounds of mitosis, 8 different types of cells. So we'll have our 6 cells on the periphery, and the cells on the other cells on the periphery, the one that matters the most is this guy here, which is our egg cell. So we have 6 cells on the periphery. The one that's kind of closest to where the pollen tube is going to enter in the center is the egg. And then they have a large central cell here. And this large central cell actually has 2 haploid nuclei. And that is what will later go on to become the endosperm. So just as a reminder, we have our diploid megaspore mother cell, then undergoes meiosis to become 4 haploid megaspore cells. And then of those 4 megaspore cells, only one of them goes on to undergo mitosis, and it gives you 7 different cells. and there are 6 haploid cells on the periphery. One of those 6 cells on the periphery is our egg cell, our haploid egg, and then they have a large central cell that is contains 2 haploid nuclei, and that will later become the endosperm. So now we have our pollen grain and our ovule containing our egg and our endosperm. What happens next? Well, next we need pollination, and pollination is just the transfer of pollen grains from the anther of one plant to the stigma of another plant, and this within the same species. Pollination can lead to fertilization, but pollination is not fertilization. So in order for fertilization to occur, pollination has to happen, but pollination doesn't necessarily mean that fertilization will occur, because you can imagine if you have pollen grains that stick to the stigma of a different species of plant, or even the same species of plant, doesn't guarantee that its gametes will actually get to the egg and things like that, or even that it even beat another plant. Another plant's gametes could have gotten there first. So just keep that in mind, that pollination is not fertilization, but in order for fertilization to occur, pollination has to happen. A variety of mechanisms can be used by plants for pollination. In the majority of time for angiosperms, they use pollinators, but they can also use wind and water as well, but not to the same extent that gymnosperms do. In angiosperms, they mostly use pollinators to transfer their pollen grains between plants. Pollinators can be a variety of types of animals. They can be birds, insects, mammals, reptiles. It all just depends on the plant species. But plants have very specific ways that they attract their pollinators. So they'll have different visual cues. So we talked about petals and how petals can be a particular color, size, and shape, and that's all to attract pollinators. They can also have olfactory cues, so they'll give off particular smells to attract their specific pollinators. And they can also have tactile cues, so they can look like they have a particular texture, or when you touch them, it can be smooth or furry or rough. And that's also a mechanism of attracting particular pollinators for that plant. A plant can have more than one pollinator. Some have just one, and they only have one animal species, or insect, bird, whatever have you, that it facilitates their pollination. But many plants can use more than one animal to transfer their pollen. And then plants can also use bribery or trickery to entice animals to come so they can use them for pollination. And if you want more details about that, see the first lecture that we did on plants. There's a strong coevolution between plants and their pollinators, and for more information on that as well, revisit lecture 1 on plants. And so now that we get pollination to occur, in order for a actual embryo to be made and the seed to be made so that you can get a viable offspring, double fertilization has to occur. So we're now going to talk about double fertilization in detail. Okay, so double fertilization, and here's where you might see some key differences from things that I mentioned in the first lecture regarding double fertilization. So take note of the steps that I'm telling you here in this presentation. So we start with the pollen grain, and the pollen grain from the anther of one plant, let's say plant A, then travels via a bee or something to the stigma and gets deposited on the stigma of plant B. So the pollen grain, which contains 2 haploid cells, one that will later become the pollen tube, and the other that will later give rise to the sperm. Okay, so it sticks to the stigma. If that pollen grain comes from a plant of the same species, then it will continue to stick. If that pollen grain comes from a plant that's a different species, then it'll basically be kind of brushed away and allowed to leave. So that way you don't get potential wasted fertilization. Well, it won't even fertilize, but you don't waste time with pollen that's from a different species altogether, because you will not end up with a viable offspring. So then let's say this pollen grain comes from the same species, we're all good to go. Then the pollen tube will start to form. So This image here shows you the pollen tube extending from the stigma down the style, and as it's extending down the style, the cells within the ovary are checking to make sure that the genetic information that will come from this pollen grain is different enough from the plant that contains the ovary. So we want to enhance genetic diversity. If the pollen tube's genetic information is identical or very, very, very similar to the genetic information of the plant that contains the ovary, then the pollen tube will start to be disintegrated by cells within the ovary to prevent self-fertilization, but to also enhance the chances that another pollen grain from a vastly more genetically different cell will have a chance to stick and fertilize the egg, giving you more genetic diversity. So let's say in this case, the plant is genetically different enough and it gets accepted, so the pollen tube will continue to extend, and the second cell within the pollen grain will travel behind the pollen tube, and as it's traveling down the pollen tube, it will undergo mitosis to form two haploid sperm cells. So now instead of one haploid cell traveling down the tube, we now have two haploid sperm cells. And once they get close to the ovule, they will enter the ovule, and one of those sperm cells will fertilize the egg to make a diploid zygote, and the other sperm cell will fertilize the endosperm to become the central cell, and that will become the triploid endosperm. So I mentioned a major drawback to asexual reproduction, but a major benefit to sexual reproduction is genetic diversity. And that's because genetic diversity gives your species a better chance at having more longevity over time. So plants want to have genetic diversity, and in order to do this, they must limit the chances of self-fertilization. And there's a couple of ways that plants do this. We just talked about one where if the pollen tube is extending and the genetic information from that pollen tube is too similar to the genetic information in the style, then the style will begin to disintegrate the pollen tube. And this is because ultimately that would mean if the genetic information is too similar, that the gametes from the sperm and the egg, once they meet, they will not be very genetically distinct from the parent plants because they carry gametes that are pretty similar. So that's one method that plants can use, but they can also use a couple other methods. Another way is they can spatially separate their male and female gametes. So in the case of corn, I don't know if you guys have ever seen corn. I'm from Indiana, so I've seen lots of corn. But at the top of corn, you have this kind of like bristly structures at the top of the very, very top of the cornfields. You can see them. And this is actually where you have your male flowers. So they're flowers that just have the male structures, and they produce the pollen grains. And then below, the part that we normally associate with being corn, which is down here, are the female structures, which houses the egg and things like that. So you decrease the chances of self-fertilization if the anthers and the ovaries are and the stigmas are spatially separated. You can also decrease the chances by alternating the maturation of the male and female reproductive structures. So some plants will have the male reproductive structures mature first and release their pollen, and then the female reproductive structures will mature. And this prevents the chances of the pollen from your own plant sticking to the stigma and trying to fertilize yourself, because the stigma is not even mature enough to allow for pollen to even stick. So this is another way. And geraniums, these beautiful plants, they are one plant that uses the staggered maturation of their male and female reproductive organs to reduce the chances of self-fertilization. And then you can also have plants like willows that have male and female reproductive structures on separate plants altogether. And so these are an example of a plant that would not be hermaphroditic. They would be dioecious, which is the term when you have male and female reproductive structures on two separate plants. So in the cases of like the geraniums and in the case of corn, they are hermaphroditic, they're monoecious. But poplars, willows, and things like that have their male and female reproductive structures, so their male and female flowers on completely separate plants, they're dioecious. And so you can see that here in this picture for the poplar. This is a example of the female flower. So I'll have the stigma, the anther, the stigma, the ovule, the ovary, all that stuff. And then this is an example of the male flower, and it'll have the anthers and the filaments. So they're on two separate trees. Now that fertilization has occurred, what happens next? Well, now we need to be able to get our offspring into its own life. We need to be able to be moved efficiently and safely to another location where it can establish its own life. In order to do this, plants use seeds and fruits so that they can protect their embryo and provide it with the nutrients it needs to have the best chances possible of survival. So seeds, as we mentioned before, but just as a reminder, are composed of a protective coating, an endosperm, which is the nutritive pack for the seedling, and an embryo. So how are seeds produced? We start right after fertilization. So after fertilization, the diploid zygote, which was a result of the egg and the sperm meeting, will undergo rapid mitosis and to form your embryo. And additionally, the endosperm, the triploid endosperm, which was the central cell that had two haploid nuclei that met with the haploid nucleus of the sperm, will also undergo mitosis to form our endosperm. As they're undergoing mitosis, the embryo will start to develop its meristem. So it'll develop a root meristem and a shoot meristem. And as a reminder, the meristems are these areas of concentrated, undifferentiated cells. And if you don't remember it in a lot of detail, we'll go over those in a second. But just remember these areas of highly undifferentiated cells that facilitate the growth of roots and shoots. And they will also, the embryo will also start to develop its cotyledon, so it's embryonic leaves. As all this process is occurring, there are also changes in the ovule. So the outer coating of the ovule will begin to harden to become the seed coat. So this whole area here will harden up and kind of thin out and become the seed coat. So then once all of this has occurred, now we have our seed is almost ready to go. But one more thing has to happen. The seed must dehydrate, and this helps to make the embryo and endosperm and everything just dormant. So they'll just be dormant within the hardened, dehydrated seed until whatever conditions need to be met for that seed to germinate. So just a reminder, so we can go over and review it. After fertilization, our zygote, our embryo, will undergo rapid cell division and will develop root meristems and shoot meristems and cotyledon. And our endosperm will also undergo rapid cell division, mitosis, to give us more and more endosperm, so that the embryo, once it germinates, once it leaves the seed, it will have enough nutrients to survive until it's able to undergo photosynthesis itself. Additionally, the ovule will become the seed coat, so it will harden and become the seed coat. And once all of those conditions have been met, the developing seed will dehydrate and will become a mature seed that's ready to go. Plants can also use fruits to help facilitate the spread of their seeds. So fruits are very enticing, especially for larger organisms like mammals and birds. So they will eat the fruit. When they eat the fruit, they eat the seeds. The seeds will pass successfully through their digestive tract and do that seed coat and be released into the feces. And then that's actually beneficial for the plant because when the plant's ready to germinate, it'll have all the nutrients it needs from the feces to have a good start to its life. All fruits are derived from flowers, and so this should make sense at this point because the fruit is the ovary. So the ovary, which is this outer portion here, will grow and grow and grow and get packed with more and more sugars, and that becomes the fruit. So every fruit came from a flower. The fruit helps to aid in dispersal, like I just mentioned, and it helps to protect the seed. It can help by, like we just talked about, animals eating the fruit, and they go somewhere else and they poop, and now the seed has been dispersed, and it also gets some nice fresh manure. But also, some fruits are not necessarily for eating. They're not palatable in that way, but they're still fruits that help with dispersal. So they can help with wind dispersal, also with water dispersal. It just depends on the plant and the type of fruit it makes. Fruits generally ripen only when the seed is mature and ready for dispersal. So I say generally because we as humans have changed some of this in plants that are agriculturally desirable, but under normal natural conditions in nature, fruit only ripens once the seed has the best chances of survival. So the seed must be mature first, and then the fruit will ripen to attract whatever animal it needs to help disperse its seeds. So fruits ripen when the seed is mature. So once a seed has made it to a new location, what happens next? Well, if the conditions are just right, then the embryo needs to leave the seed to become its own plant. And this process is called germination. Some seeds must undergo very harsh conditions in order for germination to occur. So like we talked about the example of a seed going through an animal's digestive tract, that's a harsh condition because the seed has a chance of being chewed up. It also has to get through the digestive juices of the animal and the intestines of the animal. So those are harsh conditions. In addition, some plants need their seeds to be exposed to fire before they germinate. In both of these cases, these conditions may seem kind of crazy, but they are always conditions that need to be met first to give the embryo the best chance of survival. So there are reasons behind why some plants require their seeds to kind of go through it before they will be able to germinate. So what are the steps of germination? First, let's say we have a seed that's finally gotten into the perfect conditions. There's the right amount of water in the soil, the perfect temperature, and the just right amount of oxygen. That embryo needs to now leave the seed. In order to do that, the seed, which was dehydrated, will start to uptake water from the soil, and this will cause the embryo to expand. As it's expanding, it will be pushing on the seed coat, and the seed coat, as long as it's weak enough, will rupture, and that will release the seedling from the seed, or embryo, you can say, from the seed. Once it leaves the seed, the seedling will start to grow. So this growth initially is fueled by the endosperm because they're not undergoing photosynthesis yet. So the endosperm will act as the nutritive source to allow for the cell replication of the seedling. The seedling will then extend roots downward into the soil and will start to extend after that will extend its shoots upward towards the sky, so towards the surface and then towards the sun. And then this is all still fueled by the endosperm. So the starches and fats in the endosperm are still fueling this process until the endosperm is basically run out. And then once the endosperm has basically been used all up, the cotyledon will start to undergo photosynthesis. So by this point, the cotyledon should be ready and mature enough to undergo photosynthesis, and that will start providing the starches, the sugars that the plant needs for it to continue to grow more and more and more. So this is the process of germination. The next stage in the plant's life, now that it's a seedling and it's germinated and it's starting to grow and undergo photosynthesis, is continued growth. So the plant needs to continue to grow and grow and grow as much as it can for as long as it can. Plants are unique in the way that, or different from animals, I should say, in the way that it will continue to grow for their entire lives. So as long as the plant is alive, it will continue to grow. This is different from animals. Animals generally will grow to a certain point and then we mature and then we stop. And then we continue to live, but we stop growing. But plants can continue to grow. And we see this in trees that are thousands of years old that are still growing to this day. Also, another difference from plants and animal growth is that plants, as they continue to mature, older plants are primarily consisted of old and dead cells. And we'll talk about why that is in a second. So there's two types of growth that plants undergo. They undergo primary and secondary growth. And in both cases, this is due to meristems. So in primary growth, or growth taller, longer, is a result of apical meristems. And then secondary growth, which is the girth or the width of the plant, is due to lateral meristems. And the meristems, as a, just another reminder, are these areas of undifferentiated cells that will continue to divide and differentiate into whatever cells the plants need, so they're totipotent over the lifetime of that plant. Primary growth is the growth that allows for the extension, the vertical extension of the plant. So it allows the shoots to grow taller and the roots to grow longer. It also allows for branches to form. The primary growth is facilitated by apical meristems, and apical meristems are present on the tips of the roots and the tips of the shoots. And they're essentially just unlimited sources of new cells. These cells within the meristems undergo mitosis to form whatever types of cells the plants need. So when a meristem cell undergoes mitosis, we start with our meristem cell, and it'll give rise to two daughter cells. One of those two daughter cells will also become a pluripotent meristem cell, so it will just basically become another source of differentiated cells later on. But the other cell will differentiate into whatever type of cell the plant needs. So they can be cells that become part of a bud, leaves, branches, whatever the plant needs, that second daughter cell will become. And the fact that these meristem cells are always producing other meristem cells, they're always making more of themselves, is why they're a perpetual source of new cells. So let's erase this. Okay, so as mentioned, that primary growth can lead to branching. So as the apical meristem is continuing up as tall as it can possibly go, there are kind of these segments, regular intervals, where they'll leave these notches. And these notches are concentrated areas of meristem cells. And these meristems, like clusters, later can undergo rapid cell division and form branches. And so you can see them sometimes as buds on a stem. that when it kind of has first moved past where the meristem is going to be, and then later as you keep watching, you might start to see a branch form from that lateral bud. And so you can even see on the branches, so they're showing you in this image here, even as the branches are extending from their meristems, they will also leave behind buds at regular intervals that can later become branches. So these buds are different from the axillary buds. So we also have, and you can look in this branch here is a really good example. So we have our main stem, which is this long structure here, and then we have a branch or maybe something that leads to a leaf, but some sort of branching point. And then we have this little guy here, and that's the axillary bud. And these buds basically remain dormant, and we'll talk about why that is in a second, but they basically are there just in case. And the just in case is if the apical meristem gets cut off. So if the apical meristem is removed, then that axillary bud will start to undergo rapid cell division and branch off and become a new branch and have its own meristem and keep extending. And so this ends up giving you fuller plants rather than just taller plants. And we use this a lot in, like, if you ever are into horticulture and trimming bushes and pruning bushes, a lot of times we'll do this to, we'll trim the outside of bushes, and that'll stimulate these axillary buds so that you end up with fuller bushes. Because if you have a plant that's constantly growing up but only branches a few times, you can see how slim that plant is. But let's say instead of doing that, I take this plant and I prune it. So I cut off the apical meristems up at these top sections. Okay, so I prune that bush down. Now the axillary buds that were present at all these branch points will start to grow, and I'll end up with a far more fuller bush than I started with. People use this a lot to end up with those nice full bushes you see. It's because of all this trimming. The other type of growth that plants undergo is secondary growth. And secondary growth is what results in thicker branches and trunks and sturdier branches and trunks, because it's not vertical growth. It's not growing taller. Instead, it's growing wider and thicker. And this increased width is a result of growth at the lateral meristems. And the lateral meristems, like the apical meristems, are areas of concentrated, undifferentiated cells that can go on to become whatever cells that the plant needs. And there are two types of pluripotent cells in the stems and trunks. The first we're going to talk about is the vascular cambium. So these cells, these pluripotent cells that make up the vascular cambium, are, in this image you can see here in this cartoon, they lie between the xylem and phloem in the stems. So the vascular cambium in this image is this orange ring here, and it's between the xylem, which is these blue in this image, and then the phloem, which is the pink in this image. And so as the cells in the lateral meristem are undergoing cell division, they will form more xylem, which are called secondary xylem, and the secondary xylem will be pushed towards the inner part of the trunk, towards the pith. So you can see in this image they're being produced and being kind of pushed more towards the pith, the center of the stem. And then the secondary phloem are being made, and they're being pushed towards the outside of the stem. And so as this process occurs, you'll end up with more and more layers of xylem being pushed towards the center of the branch. And you can't continue to do that forever, right? It can only become so compact. So instead of becoming more and more compact, what instead happens is the branch becomes wider and wider and wider. So you can imagine in 1 year, say in year 1, your vascular cambium is located here. And after a year of growth, your vascular cambium may be here. And after 2 years of growth, it may be here. And that's because the branch is getting thicker and thicker and thicker over time. Not all plants have lateral meristems, but some do. And so for a lot of this, we're going to be talking about trees because it's one of the easiest ways to see and explain what lateral meristems are. So in trees, this type of growth results in growth rings, specifically in temperate regions. In tropical regions, you can't really see growth rings as well, but in temperate regions like a lot of America and Canada and things like that, you can see these rings within a tree if you cut it down, and you can see these rings because as the seasons change, differences in temperature result in differences in the color of the xylem and phloem because they become either the cells are more compact or less compact. So when they're more compact, it appears as a dark ring, and when the xylem cells are less compact, it looks more light. And so you can actually count the dark rings, and that will allow you to estimate the age of a tree because each ring will represent like a winter. So looking at trees, there's also another set of there's another area of pluripotent cells called the cork cambium, and the cork cambium gives rise to the bark of the tree as well as cork cells that are responsible for water retention and protection of the sensitive tissues that lie beneath the cork cells. So getting back to the xylem, the xylem over time will continue to build and build and build. So these are the rings we were talking about, and they're building and building over time. And in this image, actually, the lateral meristem for the vascular cambium is actually way out here, close to the bark. It's very, very thin, and the phloem are lined between that lateral meristem, which I can't even draw on this image because it's so small in comparison to how large that stump is, and the bark. And so all of this area in the middle here is xylem. And you can see it looks like there's two separate colors, and that's because there's two types of wood here. So all of this is wood, all of this is xylem. Xylem. This darker area of wood are xylem that basically become clogged over time. They're the oldest xylem, and over time they've been clogged with nutrients and salts that didn't dissolve properly or built up, as well as different saps and things like that. They become clogged. So they're no longer transporting water from the roots to the shoots anymore. And they're basically just there providing structure and support for the tree more than anything. So they're very important because they're providing structural support even though they're not transporting water and nutrients anymore. This is referred to as the heartwood. And then you have your sapwood, which is here the lighter region of the wood, and the sapwood is the xylem that are newest, and they're still transporting and conducting water from the roots into the shoots. So they're all dead cells. So remember, all the xylem are dead cells, but the difference is the cells that make up the heartwood are clogged. They're no longer conducting water and nutrients, whereas the cells that make up the sapwood are newer cells in comparison to the heartwood cells, and they're not clogged yet, so they're still able to transport water. As I mentioned, we have our cork cambium. In this image, you can see the cork cambium is this nice minty green color, and it produces the cork, which then leads to the bark. So this whole area of bark is responsible for protecting all of those sensitive tissues that lie within it, but I guess beneath it. And then the cork cells are responsible for helping with water retention and protection. And they're all derived from the cork cambium. Beneath that, we have our secondary phloem and then our vascular cambium that generates the secondary phloem, and then our secondary xylem that has our heartwood, our older xylem, and then our sapwood, our newer xylem. So just being familiar kind of where these structures are is important. The phloem is removed over time, so it doesn't build up like the xylem does. So the xylem will continue to build up throughout the entire life of the plant, but the phloem get removed and replaced over time because remember those are living cells. So if they're no longer doing their job, they're replaced with new cells that will do their job. So they don't build up the same way that the xylem does. So switching gears now from growth and reproduction, we're going to talk about hormones. And hormones are just chemical signals that help to drive particular responses in an organism. And so in plants, they use hormones to regulate their growth and development, as well as their responses to stimuli and physical changes in their environment. So plants can't move around. They have to find some other way to respond to their environment, and hormones allow for the proper signaling for them to be able to respond to their environment. Hormones in plants, unlike in animals, can be produced at a variety of locations. So hormones in plants can be produced in the leaves, in the roots, in the stems, and the buds. But in animals, we produce hormones in one part of our body that get transported elsewhere. So plants can produce hormones in various structures all over their anatomy. Plants have 5 types of hormones, and they include gibberellins, auxins, ethylene, abscisic acid, and cytokines. The first category of hormones we're going to talk about are gibberellins, and gibberellins play most of the major roles in cell division and cell elongation. They're present in all structures of the plant, but in highest concentrations in the seeds and in the apical meristems. And this makes sense because in the apical meristems, we need a lot of cell division so that the plant can continue to grow and branch. And then in the seeds, the seedling needs to undergo a lot of cell division as well in order to grow and mature. So gibberellins play a major role in both of these areas and in these processes. So gibberellins play a role in seed germination because they allow for the endosperm to be metabolized. Without gibberellins, the developing seedling will not be able to access the nutrients it needs to grow and develop. Additionally, gibberellins play a role in stem elongation. The amount of gibberellins that's present will dictate the distance between the nodes on the stems and branches. So will dictate kind of how often we have branch points on a stem. Additionally, gibberellins play important roles in flower production and fruit production. And so humans, we've harnessed this ability of gibberellins to our benefit to stimulate flowers and fruits to be made and produced, even when the plant is not really ready for production due to the unideal conditions. So for example, we can apply gibberellins to plants to stimulate them to make fruits, even though fertilization may not have already occurred or anything like that would normally dictate a fruit to be made. So this is one way we can end up with seedless fruits, is because we're causing the cells to start, the plant to start making fruits, even though under normal conditions they would not make fruits because fertilization hasn't occurred. The next category of hormones are auxins, and auxins play a role in cell division and stem elongation as well. So they have similar roles as gibberellins, but we'll talk a lot more about auxins than we do about gibberellins in our lecture. But auxins also play a role in root formation, vascular cambium formation, and in the orientation of plants in response to environmental stimuli such as light or other organisms, things like that. Auxins are present primarily in the shoot tips and in immature tissues, and this makes sense because these are areas that are undergoing rapid cell division, and auxins often work in combination with gibberellins because you can see they have similar functions. And but there's one key difference in the distribution between auxins and gibberellins. So gibberellins, if you have a cell, are more evenly distributed throughout that cell's cytoplasm. Auxins, however, are not. Oxygen concentrations are highly influenced by factors such as gravity and light. So gravity pulls, downward, and this will cause auxins to be concentrated down towards the down, if you will, of the cell. So they'll be located kind of here within the cell. And this allows the cell and the plant in general to be able to know which way is up versus which way is down. If the oxygen concentrations are high, then that indicates that this is down, if you will, in relation to gravity. Also, auxins are their distribution is impacted by light. So if we have light coming from this direction and gravity pulling down this way, our auxins will be concentrated kind of in this region here, because gravity's pulling down, but auxins are moved away from light. And this uneven distribution within the cells is what causes plants to be able to bend towards light, and like phototropism. And we see that kind of bending and shaping of plant roots is due to this uneven distribution. As I mentioned before, the auxins and gibberellins kind of work together oftentimes, and they work to actually enhance the role of gibberellins. Auxins play important roles as well in root formation and branching, and in fruit development we can use auxins like we use gibberellins to enhance or encourage plants to start producing flowers and fruits even under unideal conditions. And the auxins also play a role, and we talk about those axillary buds and how the axillary buds are basically dormant until the apical meristem is removed. The reason for this is because of auxins. So the auxin concentration in the meristems is really, really high, and it actually exerts an inhibitory effect on the axillary buds. So as long as the apical meristem is present, the auxins that are in the apical meristem will keep the axillary buds dormant. But once you remove the apical meristem, you've removed the vast majority of those auxins that were inhibiting the axillary buds growth, and this will allow for the axillary buds to start to grow and develop. And this process is referred to as apical dominance. As well, auxins play a key role in the orientation of seedlings to light so they can grow, and also so they know which way is up and down. So seedlings are able to grow their roots down and their shoots up all the time, and that is due to auxins and that orientation and concentration of auxins within the tissues. We have some other plant hormones. One of the big ones you've probably heard about if you've ever watched an infomercial is ethylene. Ethylene is a gas produced by plants that stimulates ripening. And this is present in all plant structures, and it basically speeds up leaf aging and ripening and death, even death of flowers after fertilization. And this is because after fertilization, you don't want to keep maintaining flowers if you don't need them. The whole purpose of the flower is over. The egg has been fertilized. So you want to save that energy, and so they'll produce more ethylene to destroy the flower. The way you're not wasting energy, and you can use that energy now to cultivate your embryo to make your seed. Additionally, same with fruits. You want to hold off on ripening a fruit until the seed is ready and is mature, and then the plant will make more ethylene to cause the fruit to ripen, and will be under perfect conditions for the the animal to come and eat it. So ethylene plays a major role in this. The reason why I said you probably heard it in infomercials is because ethylene gas is also what causes your fruits at home to ripen. And so some people have come up with products that will cause ethylene gas to be basically degraded or removed, and it'll extend the life of your fruits and vegetables. We have abscisic acid, which inhibits the growth and reproduction of plants under adverse conditions. So if, let's say, I know you guys have all, if you've ever been in Georgia for an extended period of time, have seen those days where it'll be nice and sunny, and then all of a sudden it'll drop in temperature in the spring and it'll get really, really cold. It might even snow. When something like that happens, a plant might make this abscisic acid, which will basically cause the plant to stop doing what it's doing because those conditions are no longer optimum for growth. And they'll basically go dormant for a little bit of time so that they're not wasting energy trying to grow and reproduce when conditions aren't facilitating that. And then after the conditions are right again, then they'll remove the abscisic acid and then resume growth. Abscisic acid is present in the leaves, the fruits, root tips. Basically, they're almost everywhere, but they're present in these structures the most. And there's other conditions other than temperature that affect the production of abscisic acid, but I use temperature. It can also be if the roots get to an area that is too salty or too dry, or just something adverse occurs, and the plant needs to just stop doing what it's doing. It needs to just be dormant for a minute. This is the hormone that will allow it to do that. And then we have cytokinins. And cytokinins stimulate cell division. They have effects on all structures of the plant, so stem, leaves, buds, flowers, fruits, etc. And they work with auxins, so you can see that a lot of these hormones kind of work together, especially gibberellins, cytokinins, and auxins. They encourage rapid cell division for primary growth, which is what we talked about, similar functions as gibberellins and auxins as well. And they initiate branch formation from lateral buds, and they induce seed germination and slow the aging of leaves and the death of leaves. And this is actually so effective that in your book they mentioned that you can actually add synthetic cytokines to leaves that have already fallen off a tree and they're in the process of dying, and slow down their death as a result of adding this. So it's very interesting, and no plant has just one of these hormones. They all express all of these hormones at different times under different conditions in order for the plant to survive as best as it can. Hormones and tropism go hand in hand. And so, like I talked about, hormones allow a plant to respond to environmental stimuli. The response is tropism. And tropism is defined as the growth of a plant toward or away from a particular stimulus. And you can see this, you can visualize it as like bending and curving and twisting in plant stems and leaves. One form of tropism that most of you guys are probably familiar with is this, which is phototropism. Most people have seen a plant growing in the direction of a particular light source, And that's because they want to optimize as much photosynthesis as they possibly can. So photo is light, tropism, movement, move it towards the light. Phototropism is a result of uneven distribution of the hormone auxins, which results in uneven growth in the stems. So if we're looking as an example of what's happening here, if a plant is growing vertically and the sun is right above that plant, then the concentration of auxins will be highest in the apical meristems, the very, very top. So in this example here, if the sunlight source was directly above, then the auxins would be concentrated there. But as we mentioned before, auxins are heavily influenced, their distribution and concentration are influenced by gravity and sunlight. So if the light source is now moved to like the right or left of the plant, then the concentration of auxins will be redistributed so that they're more concentrated on the shadowy side of the stem. So now we have our light source is over here instead of at the top. The auxin concentration will be redistributed, so the auxins are more concentrated on this side of the plant stem, and that's because this side of the stem is more exposed to the light, whereas this side is in shadow. Sorry for my poor writing. But so this redistribution of auxins will cause the cells on the shadowy side of the stem to undergo more rapid cell division and for their cell walls to be far more flexible so they can become elongated. So the combination of more flexible cells and more cell division, so more cells being present on that side, will cause the stem to bend in the direction of the light. And like I mentioned, this is all to optimize the amount of photosynthesis that plant can undergo. And people can use phototropism a lot of times if you've ever seen lucky bamboo. So if you've ever been to a store, they'll have these potted bamboo plants that look something like they'll go up and then they kind of start to spiral. And this is due to phototropism. So in these farms where the lucky bamboo is being cultivated, essentially the workers move the light source every few days, and the plant will follow that light source as they rotate it around the room. And that's why the lucky bamboo will start to spiral upwards. So phototropism is actually distinct from, but similar to something called heliotropism. And heliotropism is when the plant follows the sunlight as it moves throughout the day. This is not a result of changes in cell distribution and elongation and cell numbers on a particular side of the plant. It's actually a response due to changes in salt concentrations within the various tissues of the plant, depending on where the sunlight is. So heliotropism and phototropism are two separate things, so just remember that they're distinct, but they're both our response in relation to the source of light. Another form of tropism we see in plants is gravitropism, and this is growth in response to gravity. And this is really important for plants, especially when they're first kind of coming out of a seed and germinating, is to know which way is up. So this lets the plant know, okay, this direction is down, so the roots always grow down, and this way is up, so the shoots grow up. And you can see this really well if you ever want to try this at home with plants if you turn them on their side. So this image here, you can see that this plant, this little young plant, has been turned on its side in this pot, and it's starting to grow. Instead of continuing to grow this way, which would be now horizontal when it was vertical, it now has changed directions so that it's growing vertical again, so that it ends up with this bend here. And that bending is also due to auxins, uneven distribution of auxins. So when the auxins are present, they're unevenly distributed, like I said, and then you can tell down from up. But if you change the orientation of the plant, now what was down is no longer down. Now that's the side. So the location of auxins will be changed as well. and will be transported so that the concentration is highest where you see down. And so in this case, what was up is now to the side, and what was to the side, which was right here, is now down. So the concentration of auxins will be enhanced in the new down position, and that will cause the uneven growth like we talked about in the stems, where we'll have more growth and then more elongated cells on the new down location, so that it will cause the stem to bend and bend towards up. So this is how we end up with a plant that does this. As well, this is why we see this in seedlings, where they will grow up, their shoots will go up into the air, is because of this phenomenon as well. The last form of tropism we're going to talk about in this class is thigmotropism, and that is growth in response to physical contact with another object. That other object can be living or non-living, like trees in a living sense, or poles, trellises, fences in a non-living sense. And this can best be seen in vines. So you can see in this image here, the vine growing around the tree. You can best see it like with this vine here, this one here, this one here. The vine plant, which is this green guy that has these nice beautiful green leaves, is actually the vine, is interacting with the tree that it's wrapping itself around. You can also see it here with this vine that's wrapping itself around another branch. The parts of the plant that are responsible for doing this wrapping, this winding, are called tendrils, and they basically just hold on to other objects and kind of help to anchor them. The tendrils are specialized structures. They can be specialized leaves, stems, or branches. And this kind of bending you see here, like this that's occurring, is also due to auxins. So the same thing we talked about before, auxins are responsible for why we can get these tendrils bending and winding around other objects. And this is because auxins that are produced on the tactile side, so auxins that are produced, let me erase. So auxins that are produced on this side of the plant are then transported to this side of the plant and will cause that bending. So auxins play a major role in thigmotropism, gravitropism, and in phototropism. Plants don't have watches or calendars, but they still know what time of day it is during the day. And this is really impressive when you're an immobile plant with no brain. And the reason why they're able to know what time of day it is because they have something called a biological clock. And essentially, plants are able to use pigment cells within their leaves to detect what time of day it is and change the expression of genes within their tissues depending on the time of day to have certain responses versus others. So a good example of this is seen in bean plants, whereas certain times of the day they will raise their leaves, and then like in the mornings and throughout noon, you'll see their leaves go up if you did a time lapse, and then towards the end of the day, the leaves will start to go down, and then at night their leaves are all the way down. And this is due to differences in different genes over the course of a day. So in the morning, certain genes are being upregulated or more expressed, and some aren't. And then at night, other genes are being more expressed and some aren't. So this is a really interesting phenomenon, and I've included a lot of videos of time lapses of plants throughout the day. I think the one related to biological clocks is of morning glories and how they open and close depending on the time of day. The biological clock of plants is maintained even when you remove the environmental stimulus. So if you had a potted plant and you set it outside for a couple weeks, months, and then you brought it indoors and removed the environmental stimulus, like that has no more sunlight, you just put it in a room with a regular light, that plant will continue to undergo its same behaviors as if it was outside according to its biological clock. So that's really, really interesting, and you can tell how important it is for the plant to undergo these behaviors because it remains maintained for a period of time, even though the stimulus has been removed. And then the plants are always constantly adjusting their biological clock. Because as we know, especially if you've been up here in a temperate area or the further away you are from the equator, the length of your day changes based on the season. And so, as environmental cues change, like how much night hours and how many day hours you have in a day, the plant will adjust its biological clock as well, so that it can remain in sync with the days and how much day and night time there is, so it can keep its behaviors in sync with that. So this is really, really cool to watch, especially when it comes to like morning glories and bean plants, just seeing how fast they can change over a 24-hour time period, which you may not even notice if you're just watching them every once in a while, every day. But when you see it all together, it's really incredible. In addition to a biological clock, plants have kind of like a biological calendar. And this is not the official name for it, but essentially they can tell not only what time of day it is, but they can also tell what time of year it is. And this is important because they alter their behavior depending on the season that they're currently in. And this dictates when a plant needs to undergo flowering, when it needs to make fruits, when it needs to go dormant, when it needs to prepare for dormancy. All of that type of stuff is dictated by what the time of year it is. And this is all important because a plant wants to make sure that when conditions aren't right, that it's dormant, or when their pollinators are most active, that they're producing flowers during that time or fruits during that time. So it's really important for a plant to know what time of year it is so they can effectively survive and thrive, but also pass on their genetic information to offspring, and that their offspring have the best chances of survival. So the technical term for this is photoperiodism, and it's just a plant's response to the length of light and dark in each day's cycle. So not every day individually, but as a whole. So when the days are long, well, the days are shorter versus the days are longer, will change the behaviors of the plant, because in winters, especially in temperate areas, we have shorter days, which lets the plant know it's winter. And during the summer, we have longer days or shorter nights, and that lets the plant know it's summer, in addition to temperature and other things like that. The plant knows what time of year it is and able to detect it using pigment cells in the leaves as well. And those pigment cells are mainly picking up red and far red wavelengths of light. Photoperiodism can result in kind of three categories of plants. We have our long day plants, which generally start to produce flowers in the spring. And then we have our short day plants, which produce flowers in the fall. And then we have our neutral day plants that grow and mature regardless of the length of night during the year. They produce flowers based on their own growth and maturity. So these names are actually a little bit deceptive. The plants are not based on the length of the day. The plant maturity is actually based on the length of the night. So the plants that mature and flower in the spring, the long day plants, are actually responding to the fact that there are shorter nights. And then the short day plants know that it's fall and it's time for them to undergo flowering and maturity and things like that, and reproduction, when the days are shorter, aka the nights are longer. So this is all based on nights. When the nights are longer, the short day plants will grow and they'll flower. And so around this time, things like pumpkins and apples and all that stuff, short day plants, because they have longer nights. Long day plants in the spring, tulips and things like that are long day plants. It's because there's short nights. So it's kind of confusing. That's why I kept reiterating it. Just remember that photoperiodism is based on the length of the night, not the length of the day. And just briefly, plants are able to defend themselves. We've talked about a lot of this before, so I would say I recommend going back to our first lecture if you want some more details. But basically, plants are able to defend themselves even though they're immobile. They can use mechanical defenses like thorns, spines, hairs, waxes, and they can also move. So I included a video of this plant here at the end of this presentation, and if you touch the leaves of that plant, it will curl its leaves up, and that movement is to help reduce the surface area that they can be basically chomped on by a predator. So if they're like a insect lands on those leaves, they'll curl up so that those leaves are more protected from predators. So those are mechanisms of mechanical defense. They can also have chemical defenses. This is through the production of secondary compounds like cyanogenic glycosides, which are compounds that once a herbivore eats them will be converted to cyanide, which can ultimately kill the animal or make them very sick. So it's another way of fighting back. Hallucinogens, poisons, irritants, capsaicin, which a lot of people love. I don't. I'm a wimp. I don't do spicy food. But capsaicin is actually a defense, so most herbivores would eat something that's spicy like that and they wouldn't eat it again. But humans, we just don't learn our lesson. And then we have antibiotics. So antibiotics are a way of plants fighting back against microorganisms, and that we use them to help us. Also, plants can use mimicry as a form of defense, so or basically to protect themselves. So if you look at this picture here, I wonder if you can spot the plant. The plant is actually here, and it's basically used camouflage to blend in with all the rocks in its surroundings, and it's also kind of mimicking a rock in several ways. So plants can use that, and they can also use animal defenders to help protect them. So some plants have a mutualistic relationship with other insects or animals, and it benefits each other. So the plant, the animal usually will receive some sort of nutritive source or housing from the plant, and then in return, the animal will protect the plant from herbivores or other predators. And acacia trees are a good example of this. They have these thorns, and they have a relationship with these ants, and the ants basically live in these hollowed out thorns. They literally are kind of perfect for the ant. And then the acacia tree benefits because when an herbivore or other insects come by and try to eat off of the acacia tree, these ants are mobilized and they just straight attack whatever predator that is. So it's a really good mutualistic relationship for both the plant and the animal. Alrighty, you made it to the end of plant lectures. I would say go ahead and read chapter 20. It's pretty much also almost all of 20. 20 sections 20.1 to 20.7, as well as 20.9 to 20.24, and then the chapter 20 learning curve. And then I have a ton of videos here for you guys to help you because I'm very visual, so I like to include these videos, and some of them are explaining like double fertilization with some visual aids, pollen grain formation, pollen tube formation, germination, and then I have a whole bunch of videos of time lapses of plants interacting with the sun, or interacting with each other, or their effects based on the day and night cycle. So just take a look at those if you want to. They're mostly pretty short. I think max 4 minutes at best. So if you need any further clarification, or you're very visual like me, those types of videos might be helpful. All right, excellent. With that said, we're done with plants, and we're now going to be moving on next week to fungi.